Hello, this is my video essay, and we're going to start by talking about smell. So here is our nose, and in our nose we have the olfactory tract and the olfactory bulb. So if we zoom in on it over here, we have our mucus lining. So when you inhale, airborne molecules enter your nasal cavity and dissolve into the mucus lining. Then these molecules bind to olfactory receptors located on the cilia of olfactory sensory neurons. Now, each receptor is specific to certain molecules, and this binding initiates a signal transduction pathway, where it just converts that chemical signal into an electrical one. This electrical signal travels along the olfactory nerve up to the brain. This is where it's interpreted as a distinct smell. In terms of taste, we have taste buds on our tongue and gustatory nerves. So these gustatory cells are sensitive to one of the 5 basic tastes. These are sweet, sour, salty, bitter, and umami. When food molecules dissolve in saliva, they interact with these cells, triggering a signal transduction pathway. The resulting electrical signals are sent via cranial nerves to the brain, where we perceive them as specific tastes. Now let's move on to sound. So sound waves enter the brain, and they enter the outer ear, travel through the auditory canal to the tympanic membrane, or the eardrum, located right here. This causes a vibration. As the tympanic membrane moves, it will move these 3 ossicles right here. 1, 2, 3. These ossicles are called the malleus, the incus, and the stapes. So the malleus here will move, which cause the incus to move, which cause the stapes to move. And these ossicles, their whole point is to amplify the vibrations and convey them here to the oval window of the cochlea. And this big structure right here, all of this is the cochlea. So vibrations at the oval window create waves in the fluid-filled cochlea. These waves travel through the cochlear duct, causing the basilar membrane to move. Movement of the basilar membrane causes the hair cells in the organ of Corti to bend. So if we zoom in here we have our basilar membrane at the bottom, and here we have the perilymph, the scala vestibuli, the scala tympani over here, and here is our endolymph, or our scala media. So the vibrations at the oval window create waves in all of this fluid, and then this causes the basilar membrane to move. Movement of the basilar membrane here causes these hair cells right here, to move. This bending, you see these hairs right here, these hairs will bend. And when those bend, they have mechanoreceptors that will open and allow the release of neurotransmitter after they depolarize. The neurotransmitters stimulate the auditory nerve fibers, which will generate action potentials down the nerves right here that will lead to the brain, where they're interpreted as sound. Moving on to the eye, we're going to discuss how our eyes focus light to form clear images. So, light first passes through the cornea, which begins the focusing process. It then travels through the aqueous humor over to the lens. The lens's shape is adjusted by the ciliary muscles and suspensory ligaments. For near objects that are close to the eye, the ciliary muscles will contract. This will loosen the ligaments and allow the lens to become more convex, increasing its focusing power. For objects that are further away, the ciliary muscles will relax, tightening the ligaments and flattening the lens. The retina, back here, takes that focused light and then passes it through the vitreous humor. So focused light passes through the vitreous humor, which is all this here, and strikes the retina, which is the very back of the eye. Here, photoreceptors will convert it into electrical signals, which we'll delve a little deeper into. Let's see here. So let's explore how photoreceptors in the retina convert light into electrical signals. So the retina has two kinds of photoreceptors, rods and cones. Rods, you can see in black and white, they are sensitive to low light and don't provide great detail. Cones, on the other hand, are color and they need brighter direct light to detect color. And there's three types of cones. There's red, there's blue, and green. So, when light hits the photoreceptor pigment in these cells, it causes a chemical change. That chemical change we can discuss right here. So, we have 11-cis-retinal. 11-cis-retinal is a molecule that is found inside of opsin. So, let's see. Okay. So, as I said, rods are good for low light, no color, sharp detail. Cones handle color and sharp vision. So rods contain rhodopsin, made of opsin and retinol. In darkness, retinol is bent and forms 11-cis. When light hits, it straightens and becomes 11-trans, which allows it to detach from opsin. This activates transducin. Here's transducin. Transducin is a G protein, which when it detaches, it will activate phosphodiesterase. Phosphodiesterase, all it does is it breaks down cyclic molecules, like here, where we have cyclic GMP. GMP was holding this sodium channel closed. Because phosphodiesterase broke down GMP, this channel opens, releasing a Buang down the photoreceptor. From here, the sodium rushes in. This causes calcium to rush in. You have a depolarization of the cell. Glutamate is released into the bipolar cells. The signal moves through the bipolar cells. Here we have glutamate causes mGluR6. Here it blocks this channel, causing a Buang. More release of glutamate into AMPA. Sodium rushes in, and it travels down the neuron to the brain. Now, let's break down how the brain sharpens vision using receptive fields in the retina. So each bipolar cell has a circular receptive field with a center and a surround. So here you see the receptive field center in blue, the receptive field surround in orange. So on-center cells are excited by light in the center. They use mGluR6 receptors that are inhibited by glutamate, so they activate when light reduces glutamate. Off-center cells are excited by dark in the center. They use AMPA receptors that are activated by glutamate, so they activate in the dark. Horizontal cells receive glutamate. Here's a horizontal cell. They receive glutamate from surround photoreceptors and release GABA onto the center photoreceptor, reducing its glutamate output. This amplifies the contrast, so light in the center and dark in the surround makes for a stronger signal. See, and then let's wrap this all up with a full walkthrough of the mechanics of hearing. So as we said, vibrations travel from the tympanic membrane, right here, from the tympanic membrane to the ossicles. Let's just erase here. From the tympanic membrane to the ossicles to the oval window, and then this allows a strong signal to enter into the cochlear fluid. Once that fluid movement vibrates the basilar membrane, depending on frequency, it will be a different region of the basilar membrane. But here, this movement of the basilar membrane causes the hair cells to move. Outer hair cells here will amplify that movement via Preston. Preston are channels that don't work, and what they do is they cause this hair cell to contract. So as the basilar membrane is moving, Preston causes the hair cells to move, which is amplifying the movement. Here, the inner hair cells release glutamate, and this sends signals to the brain. So louder sounds equals more vibration, more ion channels open, more neurotransmitter, more action potentials. The pitch tells you which part of the basilar membrane is active, is having resonance with the sound. And the loudness is about how many action potentials fire. Efferent signals from the brain can dampen hair cell activity, helping you tune out background noise or sleep through sounds.